Hello, good evening, everyone.
Good evening, sir.
Good evening, sir.
I hope you are doing well.
Yes, sir. And you?
I'm fine. Please, can you see my screen?
Yes, please.
Thank you. So last week, we got to look at some groups of microorganisms and their general characteristics. So I think we got to protozoans. Does this slide look familiar? Have you gone through the slides? Any confirmation, please?
No, sir.
Hello? Anyone to confirm? Are you there? Can you hear me?
We can hear you loud and clear, sir.
So, okay, somebody is saying go to viruses. So that's after protozoans. All right, so we thought about viruses as acellular entities. They are not living organisms because they lack cells. But these viruses can cause infections and diseases because they are intracellular parasites. They need a living cell before they can replicate. And in the course of replication, they end up causing harm within the host. Some of them can lead to organ dysfunction, disruption of the tissue functions, and so on and so forth. Some of them hijack the cell and end up killing the host if treatment is not given promptly. So the one who cannot hear, check your volume. Mine is at 100, so check your end. Someone is also saying we didn't look at protozoans. So Karen, does this slide look familiar? This one, does it look familiar? Do you recall, Karen? What about this? Where we looked at the subgroups under protozoa. Please do I agree with Karen? It seems this looks strange to her. All right, so I'll take the protozoans again. But for the fungal pathogens, we looked at them. So for protozoans, they are unicellular. They have only one cell. unlike other fungi that are multicellular. Protozoans are unicellular, and they are relatively larger than the bacteria. So some can be as large as 100 micrometers. So if you are to convert micrometers to millimeters, how many micrometers will make 1 millimeter? So those who are saying my voice is low, please check from your end. Check the volume from your end. Mine is at 100. So if I shout, it's going to disturb the others. So how many micrometers make 1 millimeter? Make 1 millimeter, yes. How many micrometers? Do you remember the scale of measurement? All right, so 1,000 micrometers will make 1 millimeter. So if you divide 1 millimeter into 1,000, you get 1 micrometer each. And for protozoans, they mostly prefer aquatic or water environments, although some can dwell in areas that are quite drier, but they prefer aquatic environments. And they possess appendages that can help them to move, so they are motile. Because they are eukaryotes, they have nucleus, but they are unicellular, they are single-celled organisms. They also have other membrane-bound organelles, like the mitochondrion. And in terms of feeding, they can surround the food material or the products and then take them in to digest. Some of them have means of taking the nutrients directly, so they are absorbers. A few are photosynthetic, but we shall focus on the ones that cause infections. Those that absorb nutrients directly from the environment could be the host. So once they find themselves with the host, they can directly absorb nutrients from the host and then multiply. And those are the parasitic ones that can cause diseases. So Joyce, is the voice okay? All right. So we can put protozoans into subgroups based on their features or characteristics. We can have those that are amoeba-like, the sarcodina. So example is the entamoeba species. They infect the intestinal tract. We have those that have flagella that move by means of the flagella. So they are the flagellates, and scientifically we call them zoomastigophora. Some of them can infect the intestinal tract. Some of them can infect the genital areas. And some of them can cause blood infections, as well as the tissues. So we can have Giardia lamblia, Trichomonas vaginalis, Trypanosoma species, Leishmania species. For the ciliophorans, they also have cilia for movement. Example is the Palantidium coli. That can also infect the intestinal tract. the apicomplexan parasites or the sporozoans, they form the sporozoites, and these can infect the blood, tissues, and lungs like the pneumocystic carinii. Some can infect the intestinal tract like the cryptosporidium that causes cryptosporidiasis. For malaria, they are under the epicomplexans, the plasmodium parasites. So malaria is not caused by mosquitoes, but caused by plasmodium parasites. They are rather transmitted by mosquitoes, the female Anopheles mosquito. So we have several protozoans that can cause diseases, and they can cause disease in various sites of the body. Some can be transmitted by sexual means. Some can be transmitted by the fecal-oral route. So contaminated food, water, these can serve as source of the infection. Some can be transmitted by vectors like mosquito, the sandfly. So protozoans like the Trichomonas vaginalis, Entamoeba histolytica, Diarrheal amblyii, or duodenalis, Cryptosporidium, Plasmodium, there are many. But these are the common diseases that we can have from these protozoans. And like I said, they have appendages that enable them to move and then to better live in the environment and then multiply. But they are all eukaryotes, so they have nucleus. So nucleus is prominent in all these. They also have other organelles that are membrane bound, like the mitochondrion that is present in all protozoans. This is a typical Plasmodium parasite that has nucleus, and they have membrane coat. So these merozoites surface proteins are highly variable. Sometimes the immune system can use these surface proteins to recognize the Plasmodium parasite and attack them by means of defense. Sometimes we can also use these proteins, not only the surface proteins, but other proteins like the circumsporozoid protein, to produce vaccines against these Plasmodium parasites. But because these proteins are highly variable, they keep changing, and if one particular protein is changed, and that was what the researchers used to produce the vaccine, it's likely that if the new strain infects, or another person gets exposed to that particular new strain that has changed, the vaccine might not work. So for vaccines that are targeting Plasmodium, you don't always have 100% potency because of these challenges. They keep changing. The parasite keep changing. But aside vaccines, there are other medicines that can be used for malaria treatment. Vaccines are only there for prevention. For ailments, these are the parasitic worms that we shall focus on, but there are numerous of the non-parasitic type, and these, some of them are larger, they are macroscopic. they can be seen with the naked eye, but because they also cause infections or diseases, we still include them as part of microbiology, the study of microorganisms. The techniques that are applicable to these tiny organisms are also applicable to these worms. That's why we study them as part of microbiology. So for helminths, we can have the nematodes, the cestodes. The nematodes, we have the Ascaris, Lambrecoides, Trichuris, Trichura, hookworm. The cestodes, we have the Hymenelepis diminuta or the Nana, the Antinia species. In terms of size, they are not as large as the protozoans, although some of the helminths could be very large. So for viruses, these are not part of the principal groups of microorganisms, but because they cause infections, we include them in the study of microorganisms. And viruses are complex macromolecules. It doesn't mean they are macroscopic. Macromolecules are the carbohydrates, the proteins, the lipids. So because they either DNA or RNA, we say they are complex macromolecules. They also have proteins, but they are not macroscopic. They are very, very tiny, ultra microscopic. They are not living. They don't die. They are inactivated. But you mostly hear people say the virus has been killed anyway. And viruses are species specific. So we can have viruses that infect plants. So Phenoria, your question is, do the helminths have different presentations according to their types? Come again, different presentations as in?
Please, can you kindly move to that the slide? Say, as they are here, with the way they are looking, when they infect someone, can they do they have different presentations like how the clinical presentation?
Symptoms, in terms of symptoms and signs. Is that
Yes.
what you mean? Oh, okay. So mostly
Say.
you have these infecting the intestinal tract. Some can also infect other organs. So some of these helminths, their symptoms or signs will mimic other infections. Some of them, you will have fever. Fever is not specific to any particular infection or disease. Some of them will lead to anemia. So anemia is not specific to any infection. So they share these presentations with other infections or other pathogens that also cause these. Some of them would also lead to diarrhea. For some of them, weight loss. Some of them, they could present as asymptomatic. The person could be having the infection but will not show any symptoms. But with time, the person may harbor a lot of them, and if care is not taken, can lose his or her life. Sometimes these helminths, they will also pave way for other infections to set in, because by means of weakening the immune system, certain organisms could be opportunistic So if those organisms ideally will not cause infections in healthy people, because of the presence of these helminths, they could also end up becoming opportunistic pathogens. So in terms of presentation, they are usually non-specific. And mostly you can also have co-infection where you have helminth and another bacterium or another protozoan infecting the same host. And because most of these are gotten through the soil or contaminated food or infected organisms that are eaten, you may also and usually result in co-infection. So if the immune system is strong, we usually is able to take care of them. If you know that, you're okay.
Yes, sir. Yes, sir. Thank you.
All right. So we can have viruses being species specific. Some of them can infect animals and humans. So we can have a switch from these animals and then they infect humans. Some of them will infect only plants. Some of them will infect only animals. Some of them will infect only humans. That's what we mean by they are species specific. And there are several viruses, thousands of them. But we keep researching to see the emerging ones and the ones that are changing over time. All right. Then proteins that can end up causing diseases, the infectious proteins called the prions. So some proteins in humans can misfold and this can lead to some abnormalities. So we call these prions. So for the viruses, they come in various sizes, various forms, but they are all intracellular. They need a living host, a living cell before they can replicate. A virus cannot grow or replicate on its own. A virus has either DNA or RNA. It doesn't have both DNA and RNA. Unlike all living organisms that have both DNA and RNA, viruses don't have those present in them. And the DNA or RNA is protected by a protein coat. So remember we said that they are complex macromolecules. So macromolecules include the DNA, RNA, proteins. That's why we refer to them as such. Aside the protein coats protecting the DNA or RNA, some viruses have lived viruses. They need a living cell before they can be activated. And in terms of size, they are very, very small. And some of them can cause diseases. The few that we know to cause infectious disease, viruses come after bacteria in terms of numbers. But you can have the beneficial ones, but we always focus on the harms that they cause. And in terms of size, remember we are looking at the micrometer range for bacteria, for protozoan. So they are very small, nanometer range. So the microscope does not see these viruses. The light microscope that we find in our labs, hospitals do not see these viruses because of the size, ultra microscopic. So I've talked about them being DNA or RNA in nature, and then the protein coat around the DNA or the RNA is referred to as a capsid. So when you take these protein coats, they are made up of subunits called capsomers. So the capsomers come together to form the protein coat, which we call the capsid. So this protein coat plus the DNA or the RNA of the virus is known as the nucleocapsid. So when you mention nucleocapsid, it simply refers to the protein coats plus the DNA or the RNA of the virus. And this is what we term as a viron or a complete particle. So once the host gets exposed to the virus, what the virus does is it needs to get its DNA or RNA replicated. So this protein coat, which is the capsid, will open up and then release the DNA or the RNA to the host cell, and then it takes over to multiply. Once it takes over, it uses the information within the DNA or the RNA, those genes, to control the host cell. So the host cell does not have power on its own. It is then controlled by the DNA that has the information within the virus, and then it does what it does. It likes to the host, unless there is an intervention. So maybe an antiviral agent is taken in to stop that process, or the immune system is boosted to take control. So the host cell will express the genes within the DNA or the RNA of the virus. So talking about those that have membranes and those that don't have, we can have non-enveloped viruses. So those ones have only the protein coat, which is the capsid, plus the genome, the DNA or the RNA. The enveloped viruses have the DNA or the RNA, which is the genome. So the total genes within an organism or the virus is referred to as the genome. This is the protein coat surrounding or protecting the DNA or the RNA of the virus. In addition, they have a membrane protecting the capsid. So this is a phospholipid bilayer made up of phosphates and then lipids. So usually you also find these in the cell membranes, the phosphates and the lipids. So for viruses that are DNA in nature, you know DNA is double-stranded. So there are two strands that run in opposite directions. So they are anti-parallel. And eukaryotes have linear DNA. Prokaryotes have circular DNA. For the RNA viruses, the RNA is usually single-stranded. In few cases, there can be a double-stranded RNA, but generally a single-stranded and is linear. Some of the viruses have the RNA segmented. So they can have various forms or various genes that are broken but interspersed within the capsid. And the RNA viruses could be positive sense or negative sense or antisense. So antisense is the same as negative sense. And this negative sense is complementary to the messenger RNA. So for DNA to have the genes expressed, there's what we call flow of genetic information. The DNA has to be changed to RNA, and this is what we refer to as transcription. And the RNA is decoded into amino acids. So the decoding of the RNA into amino acids is referred to as translation. So for the DNA viruses, once they hijack the host cell and they need to express their genes or multiply, they will replicate and then change their DNA to RNA, which is then decoded into amino acids or proteins. But for the RNA viruses, because they're already in RNA nature or RNA forms, they can be directly decoded into amino acids. So it takes a short time for them to express their genes or their genome. So if it is positive sense, that means right away they can be decoded from the RNA. This is what it means. Gene expression means the DNA being changed to RNA and then changed to proteins. If it is, please mute. But if it is RNA and it is negative sense or antisense, because it is negative, that means the sequence of the RNA cannot be directly decoded into proteins or amino acids. This is because it is read backwards. That's why it's referred to as negative or antisense. So there's a need for a protein. Oh, sorry, the line is breaking. Can you hear me now?
Yes.
Hello? Please, can you hear me?
Yes, but still a bit cracky.
Oh, okay. Thank you. It's about to rain, so the network is getting poor, but let's manage. I hope it's better now. All right. So I was talking about the antisense or negative sense viruses that are RNA. So these ones, the way they are the RNA sequence appears, it is read backwards. So there's a need for an enzyme called the RNA dependent RNA polymerase that needs to read or rearrange the sequence so that it can be decoded into amino acids. That's why they are referred to as negative sense. But for the positive sense, they are the amino acids or proteins for them to express that are within the virus. This is different from reverse transcription. So some viruses that are RNA can convert their RNA back to DNA with the help of an enzyme called reverse transcriptase. So like the HIV, it can convert its RNA to DNA and then replicate and then convert its DNA back to RNA and then modify it. So in the process, it can change several times. That's why it's difficult to get vaccines for HIV. These viruses keep changing. So RNA viruses like the coronaviruses, they also keep changing. As the viruses replicate or transcribe or reverse transcribe, they have some mechanisms to intentionally delete some of their sequences so that the cell gets confused in a way. So the cell gets confused so that it doesn't track them or monitor or have memory of them. And then this is how they're able to survive within the host cell. So this viral genome encodes a very few proteins, enzymes. So once the DNA or the RNA is present in the virus, it has some genes that can be used to make these enzymes, these to form the complete virus, the protein, and then the other parts of the virus, like the membranes. But all this information are within the DNA. So the DNA controls every activity within the organism or the virus. That's why when the transcription or translation process is blocked, the virus cannot multiply further or express its gene or its genome. So the virus becomes inactivated, and with time, the immune system can take over to destroy them. I hope it's clear on the viruses. All right, so for prions, you will not spend much time on the prions because they cause rare diseases. In our part of the world, we don't usually focus on these rare diseases, but in the advanced countries, once the baby is born, or even before that, they can investigate, screen, and know the particular rare disease that the baby is likely to have. Yes, Finoria.
Sir, please, I don't know if I'm alone, but then I didn't really get the senses well, the anti-sense, those senses, the negative sense, the positive sense, and so you mentioned one again too. I didn't get them well. So please, if you can take it again for me.
Okay. So to understand positive sense, negative sense, take note that it has nothing to do with charge. So forget about the positive, negative. It has nothing to do with charge. Positive and negative has to do with the direction of the RNA sequence. The RNA sequence can be read forward or backwards. RNA viruses have messages read forward and can be directly decoded into amino acids or proteins. So those ones are referred to as positive sense. They can be read directly or decoded directly from RNA to amino acids. For the negative sense, the sequence of the RNA is read backwards. That means they cannot be directly decoded from RNA to proteins. When you talk of decoding from RNA to proteins, it means that the gene is being expressed. The gene expression has to do with how the proteins are arranged. So if you have phenylalanine, serine, tryptophan, another serine, methionine, arranged in a particular order, it brings about the expression of a gene. If the arrangement changes, it also affects how the gene is expressed or the sequence appears. How they are decoded will instruct the cell to behave or act in a particular way. But for the negative sense, because the sequence is read backwards, it will be changed to be read straight away, not from the backward state. So there's a need for an enzyme called the RNA-dependent RNA polymerase that will rearrange the RNA sequence so that it can be directly decoded into amino acids. So for the RNA virus that need the sequence to be changed, rearranged, so that it can be read or decoded into amino acids, those are referred to as the negative sense or the antisense RNA viruses. I hope it's clear. Before I get to the next one, Finoria.
Yes, sir.
Yes, so I also said that don't get confused with reverse transcriptase. So reverse transcriptase is another enzyme that can convert the RNA into DNA. So conversion of DNA to RNA is referred to as transcription. So if you share RNA to reverse tribe, that's why that does it is called this transcriptase. So reverse transcriptase will the RNA back to DNA so that it can be replicated, it can be doubled, can be multiplied. And then once it is multiplied, it can then be converted back to RNA and then decoded into proteins or amino acids. And the decoding is what we refer to as translation. So in simple terms, you can have words. So let's have this analogy. If you have words, you go to the bank and then you ask to write the amount you want to withdraw in words. So you write 1,000 ONE, so 1, then 1,000 in words, cedis. Let's assume that is the DNA. The person writing it has written it in words, 1,000. Then you ask to write it in figures. Then you write 1, 0, 0, 0. So if you write in words and it is also written in figures, it has the same meaning. So DNA is changed to RNA by the same meaning. So in words and in figures. So the figures, let's have it as our RNA. Then the teller knows what you have written. So it needs to express or give meaning to what you have written and you want to withdraw. So the person will count 1,000 for you. So that is the expression of what you have written. So changing from the RNA to DNA to amino acids is the gene expression. writing in words will end up getting you 1 counted for you to take home, but it's the same expression. So this is what gene expression is about. All right, I hope it's clear. So the DNA cannot be expressed directly as a gene. The DNA cannot be expressed directly as a gene. The genes are within the DNA, but for them to be expressed, they need to be changed from DNA to RNA, from RNA to proteins. That's the flow of genetic information. All right. So if your hair will grow, the genes that control hair growth have to be changed from DNA to RNA and then decoded to proteins. If your cells will multiply, there's a gene for that. It has to be changed from DNA to RNA and then decoded to proteins. So that's how genes are expressed. All right. So prions, they cause rare diseases in humans. So it's the same protein, but depending on how they misfold, the pattern of misfolding, you can have a different name for it. So we have the Creutzfeldt-Jakob disease, fatal insomnia, and the Kuru. All these are as a result of misfolding within the proteins that are within the brains. And these disorders, though they are rare, they exist. So in terms of sizes, viruses are very small. They are not cellular. They lack cells. All other organisms, bacteria, fungi, protozoans, helminths, they have cells. For the nucleic acid, when you talk of nucleic acid, it refers to DNA or RNA. Viruses don't have both. They have either DNA or RNA. Bacteria, fungi, protozoans, algae, they all have DNA and RNA present in the cell. For the nucleus, bacteria, they don't have nucleus, so they are prokaryotic. Fungi, algae, they are eukaryotic. They have nucleus, true nucleus, so eukaryotic. Viruses don't have cells, so we don't expect them to have nucleus. Ribosomes, for cells, they don't have because they are not cellular. Mitochondrion, no. For mitochondrion, bacteria is prokaryote, so they don't have membrane-bound organelles, but they have ribosome. The ribosomes in bacteria are 70S. Ribosomes in eukaryotes, so fungi, protozoans, human beings, 80S. Are you sure we didn't do this last week? Anyway, let's continue. So 70S, S refers to the Swedberg unit. Swedberg. It's named after the scientist Swedberg. So he was able to investigate sedimentation velocity if try or push the ribosome to the bottom of a tube. What is the force needed to push this ribosome? So it refers to the density ribosome. This ribosome is made up of 2 subunits, the large subunit and then the small subunit. The large subunit is 50S, the small subunit is 30S. When the two are put together, you get 70S, not 80S. Take note. For the large subunit, which is 50S, it refers to the force needed or the sedimentation velocity needed to push the large subunit, which is 50, the 50S down. So the force needed is 50. That's why it's referred to as the 50S ribosome. The small subunit is smaller than the large subunit, of course. So the force needed to push it down when you centrifuge will be less. So that one is 30S or the 30S Swedberg unit. When you put the large and then the small subunits together. And then you set the tools that require push the respective at the force friend the force needed to push you and your friend will be different. If you are put together, the force needed to push the two of you together will not be the additional forces of you and your friend. So take note, that's why we have 70S coming from the 50S and then the 30S for bacteria. Faustina, is it okay? Is the line okay?
Yes, sir. It's okay now.
All right, let's continue. It's really, so please bear with us. And for the 80S, this is made up of 2 subunits, 60 and then the 40. So the 60 is the large subunit, 40 is a small subunit. The velocity or the force needed to push the large subunit is 60. The force needed to push the small subunit is 40. But when the two come together, and they are usually separated, they only come together during translation. That's why we say protein synthesis occurs in the ribosome. So the RNA will be brought and then sandwiched between the large subunit and then the small subunit. That's why we say protein synthesis occurs in the ribosome. So take note. When the two come together, 60 plus 40, you have 80S. So 80 Swedberg units and not 100. Take note. So all eukaryotes have the 80S. All prokaryotes have 7 chromosomes. That's the large and then the small sub. All right. So because eukaryotes like human beings and prokaryotes like bacteria don't have the same ribosome, we can use some medicines or some drugs that will target these individual subunits and not that of the humans. But when it comes to controlling fungal infections, protozoan infections, it's difficult to target the ribosome because we share the same ribosomes. So usually we don't target the ribosome when it comes to fungal infections, protozoan infections, because they are all eukaryotes. But for bacterial infections, we target the ribosome. All right. So this comparison between the viruses, bacteria, fungi, protozoans. Please, it's raining heavily here, so bear with us, the network. I hope it's better compared to the previous weeks.
Yes, sir.
Yes, sir.
All right, thank you. So this is the scale of measurement. We have the sizes reducing as you move to the left. As you move to the right, this spaces. So you can see with the naked eye. 1 millimeter. You say how many 1 millimeter? Yes, how many centimeters will make 1 millimeter? Who can tell us? How many centimeters? 1 millimeter. Yes, you can try. 0.1, good. So 10 millimeters will make 1 centimeter. Good. So if you divide 1 millimeter So you can think of the meter rule. 1 millimeter, you divide it into 1,000, you get 1 micrometer each. And this one, your eye cannot see. So you need a microscope to help in that. So the microscope sees in the micrometer range. The plant animal cell, they are within the 100 micrometer range. The fish egg is relatively larger than the plant cell, animal cell. But the organelles, the individual components of the cell, are very small. The chloroplast, the bacterial cell, very small, within the 1 to 10 micrometer range. So we cannot see these with the naked eye. But when they are multiplying, when they are growing, they form colonies, they come together. So you can have millions of the bacterial cells that are together. And that's how you can see a colony on the petri dish or the plate with the naked eye. But as you move from the micrometer to nanometer, that means you have divided 1 micrometer into 1,000. So you get 1 nanometer each. And this is ultra microscopic. So it has moved from the micrometer range and now you are in the nanometer range. So the microscope cannot see in that range. So you need another powerful microscope, the electron microscope, to see also beyond the scope of the electron microscope. So there are limitations anyway. So just imagine how small the cells look like. And that's why we need to use various means to detect these pathogens. So this brings us to microbial infections. Please, can you see the screen is moving? The slides are changing. Can you see them?
Yes, sir.
Please,
Okay.
yes,
Yes,
we
sir.
can.
So microbial infections or host pathogen interaction. So pathogens will cause infections when they interact with the host. For the normal flora, they will not cause infections unless there's an opportunity or when they move from one site to the other. So we can have microorganisms interact. It can be mutualistic. You benefit, the microorganism also benefits. Can be commensalism, where the microorganism benefits, you don't benefit. So you only provide nutrients to the microorganism, they don't harm you, you don't benefit. Some of them will benefit, you also benefit, like the ones that are in the intestines or the ones in the vagina. They can help to maintain the pH, the acidity or the alkalinity of those sites. Some of them also help to prevent other organisms from colonizing those sites. So in the mouth, you can have several microorganisms that will prevent other organisms from colonizing the sites. Some of them will produce chemicals that are toxic to other microorganisms. So these normal flora are usually helpful. So they engage in mutual interaction with us. But when there is an infection is when an organism or a virus penetrates the host defense system. So once you get exposed, that's not the end. There need to be penetration it needs to get into the cell or the tissue. So once there's a foreign agent, bacterium, fungus, virus, the host will sense that there's a foreign material, so let's attack. And these organisms or the viruses, they are also strategic. They can have a way to avoid being attacked. Some of them can cover themselves up. Some of them will have membranes that will deceive the host cell or the defense system. Some of them will prevent being engulfed because they have slimy layers. And then some of them can also neutralize the host defense system by producing other chemicals so that they can bypass the barriers that the host is using to attack or defense itself. Then they multiply in the course of avoiding being attacked, and they get to the site where they are going to multiply and then cause damage to tissue or disrupt the functions to the organs or the tissues. So infection has to do with invasion and then multiplication. So just getting exposed to the organism is not infection. It needs to invade sites and then multiply. And then avoiding by the system. Or fully supply. There are several diseases. Some of them are coming from the pathogen. So if it is responsible for the disease, then it becomes an infectious disease. Yes, it's raining heavily here. So God's love, please bear with us. But I hope you can hear me. Please bear with us.
Yes, sir.
Please, can you? All right. Sorry about that.
No, sir, we can't hear you.
I'm only hoping that with the technologies we have, anytime it rains, we will not have these challenges. The telcos have the net very bad, but this is a challenge we are facing. So please with us. So I'm saying that there are several diseases. For example, diabetes. Diabetes is not caused by pathogen. So diabetes is not infectious disease. But malaria is an infectious disease. Typhoid fever is also called, so it is an infectious disease. And these infections, when they result in damage or disruption to tissues or organs, that elicits symptoms or impairment of the functions of these organs or tissues and affects the host, we term it as infectious disease. So infection will progress to disease. Also, please, can I move on? Hello, please, can I move on? Just bear with us. This too shall pass. All right. So let's talk about flora. There are some organisms that are present on our skins. Some of them are present in the mouth, in the intestinal tract. Some of them are present in other sites like the vagina, and they don't cause harm. So they are residents. Your body knows that they are part of the system. So they don't bother to attack them. But sometimes when the levels go up or physiological processes go or change, then these ones, the body will try to react. But there are some sites within the body that should be free of microorganisms. So the organs, the internal organs, the tissues, the fluids like the blood, the lymphatic system, or the lymph nodes, all these are supposed to be free of microorganisms. The blood vessels. So anytime you have the microorganisms in such sites, infection will result. So if, for example, you have the normal flora in the intestinal tract, so E. coli could be normal flora, Proteus could be normal flora. If there is ulcer or intestinal perforation, or if someone takes medication and these wash the linings of the intestinal tract, or if somebody takes excessive alcohol and the tracts or the linings that are present within these tracts get washed off, they could perforate and get into the bloodstream, the host. So because they are normal flora, I mean they'll be normal everywhere. When they change location within the same host, they can result in infections. There are some that are not resident. They come and go. So those that are on the skin, when you bath, when you sweat, you lose these normal flora, and then with time, the environment will also bring them back. When you shed tears, you lose some of these normal flora. So normal flora starts from growing and becoming harmful to us. So they produce toxins or chemicals that will prevent these from harboring in those sites. Some of them will also maintain the pH, and that will not be conducive for the other organisms from establishing themselves. So remember, we look at the conditions or the factors that are necessary for microbial growth, and pH was a condition or one of the factors. So we have microbial antagonism. When a microorganism or the normal flora present in the host prevents other organisms from establishing themselves and then causing diseases or infections. So Maxwell is asking, is normal flora related conditions treatable? So if I get you, if they end up causing infections, can they be treated? Yes. Sometimes even when we take in antibiotics or some medicines, they can cause imbalances in the normal flora. So those of us who have been taking antibiotics anyhow, some of them can lead to imbalance in the normal flora and can pave way for other organisms from establishing themselves. The good thing is, because these organisms multiply at a very fast rate, when the levels go down, multiply and then get to a threshold that is needed, sometimes they can overgrow and result in imbalances, and then that can also affect the host, especially if the immune system is down or compromised. Yes, so these normal flora, when they cause disease or infections, they can be treated. So when these normal flora get to other sites and result in infections, they are referred to as endogenous. Endo means within. So the infection is coming from the same host, not from the exposure to another organism. The organism is already present in the host, but because it has moved from its usual site to a different site, it is causing an infection. So that is endogenous infection, like where you have the intestinal tract being harbored by resident flora, and then they can get into the bloodstream. So this is and you can also have a cut, and then those normal flora on the skin will get into the site where the cut occurred, and then they can cause wound infection. You can also have a cut from the mouth. Normal flora will get into the bloodstream and then result in endogenous infection. But the good thing is, if the person has a strong immune system, usually they are controlled or they are taken care of, and this will not will end up in infection. Usually happens in those that are having the immune system compromised. So for these microorganisms to become pathogens or cause infections, remember I said it's about invasion and then multiplication. So they need to be able to overcome the immune system, the defense system. So how well they're able to invade, it has to do with multiplying at a fast rate, having structures that can make them bind firmly to the host, structures that can make them swim or move freely within the host, being able to produce substances, toxins, that can neutralize the immune system or make them survive within the site where they are going to stay and then move to other sites. So the inversions or the invasions can be produced by these pathogens, and then they can colonize the sites and move on to cause infections and diseases. Then the severity of the disease has to do with the virulence, the structure that they have to cause the diseases. So the more virulent they are, if care is not taken, a person may lose his or her life. Some of them produce toxins, and these could be exo or endotoxins. Usually the exotoxins are released, endotoxins are not released. They are released only when the organism is by their toxins that they release within the system. So I have a question. Not only normal flora, but the usual pathogens. We have been exposed to so many pathogens, but we don't fall sick because the immune system is very strong. So you all test positive because you have been exposed to TB, You have been exposed to microorganisms. You have been exposed to viruses, so many of them. But we still look strong. So if your immune cannot bypass and then cause infection, that's a good thing. So always keep your immune system boosted. Stress can lower the immune system. So don't get stressed. Don't think about things that will make your immune system go down. Eat balanced diet and you are good to go. All right. And always be conscious of your environment. You don't know the organism that you are getting exposed to. So always be conscious. All right. So, yes, back to the question that the gentleman just asked. Oh, okay, that was Otin. All right, Prince. So, infection has to do with this stage. So, look at the stages. There should be an entry point. So, it can be through inhalation. If you are in a hospital and then patients are coming in with respiratory infections or diseases, some will be coughing, sneezing, some will be spitting, and they will be touching surfaces. So once you are exposed, you have these incubated. Then they will have structures to bind to the surfaces of the cells and the tissues. So they need to adhere firmly. Sometimes when they adhere, then the immune system will prompt the body that there's a foreign material, so attack. So they can deal with those at this site, at the adhesion stage. Some of them will successfully invade other sites initially and then multiply and then be taken care of. So the immune system will still continue to fight, even though they are multiplying. But if they are overcome, then finally they move to the sites where they are suited. So for example, for malaria, we have the mosquito taking in blood meal and then accidentally introduce the parasite. Then the parasites will move from the skin and then to the blood vessels and then to the liver cells, infect the liver cells, then come back to the bloodstream, multiplying, and then infect the red blood cells. So it depends on the entry point. Then you have disruption to tissue function, organ impairment, then symptoms are setting in, then the person begins to feel all these and will seek treatment or management. Then we'll begin to them, so can be through the same point where the infectious agent passed, or it can be a different point. So it can be through feces, through urine, through coughs or spitting, can be through blood. So there are several ways through which these can come out of the host. All right, so somebody is asking about the difference between infection and then virulence factor, if we should go back. So the ability to invade the tissue and then multiply. So the pathogen is the organism that has the ability to invade the host and then multiply. That's a pathogen. but the degree of the invasion and multiplication is what we refer to as a virulence. The severity of that particular disease. So once the pathogen multiplies, it takes over, and then definitely the tissues or the organs will be affected. But the severity of that particular disease resulting from the infection caused by that agent is what we refer to as the virulence. And this has to do with structures that they have to make the condition more severe. Say, can you say the ability of the microorganism to fight the immune system and overcome it? Severe degree of the pathogen causing the disease, that is the virulence. The degree of how the disease progresses, the severity, that is the virulence. But before this, there should be invasion and then multiplication. That is the pathogenicity. So a pathogen has to, if you should go back, invade and then multiply. To do this, it needs to overcome the immune system, the host defense system. So once it's able to invade and then multiply, it is a pathogen. So pathogenicity has to do with being able to grow and then multiply in the host. And this will result in an infection because it needs to overcome these barriers before it gets to the infectious site or the infection site or the target. To get to the target, it needs to overcome several barriers with the defense system. So that is part of these stages. But the severity of this infection, this disease, is what we refer to as the virulence. So being able to invade, multiply, and cause the infection is one. But how severe it becomes is the virulence. Do you get the difference now?
Yes, please. Thank you.
All right. And this has to do with certain structures that they have. Some of them, so where are we? Yes, so some of them have to do with the structure that they being able to take in nutrients directly or being able to produce certain chemicals that will disrupt the tissue organ function. And once these are disrupted, the disease will progress. Some of these have to do with the characteristics of the organism, what structures they have, and then how well they're able to multiply very fast and then overcome these barriers. This is why I talked about the toxigenesis. Being able to produce toxins is one of the virulence factors. All right, so talking about pathogens, those that are able to invade and then multiply. We have the true pathogens and then the opportunistic pathogens. The true ones are the ones that any healthy person gets exposed to them. So once you get exposed to any of these pathogens, exposure is not infection, but the true pathogen will invade or multiply and then get to the site. They have strategies to overcome the immune system. So if you don't disinfect, they will rather infect because they are true pathogens. If you don't prevent them, they will at all costs multiply and then overcome the immune system. Then we have the opportunistic pathogens. These ones, in a healthy person, they don't cause diseases or infection. They only cause infection when there's a problem with the defense system. So when the immune system is compromised, or when they grow in other parts that they are not found, so if the site is new to them and they get themselves there, they cause infections or diseases. So example is Pseudomonas candida. So you can have candida in the blood, but you can have candida in the vagina and could be normal. But seeing them in other sites, you can even have candida infection of the eye, candida infection in the lungs. Because they are not found in those sites naturally, they can result in disease because these are opportunistic. And this happens when the immune system is compromised. Sometimes you can have opportunistic infections, even from medication. So there are some medications that can weaken the immune system for some time, and then some organisms may take advantage. So viral load. No. Viral load has to do with the number of viruses per microliter. So usually if you are measuring the viral load, how many viruses are present per microliter? That's a viral load. It has to do with only viruses. We can have bacterial load. But virulence is the degree of pathogenicity, the severity of the disease. That is virulence. It's different from viral load. Viral load is how many viruses you have. So if it's HIV, how many of the HIV are present in the host? That's a viral load. If it's hepatitis, how many are present? That's a viral load. But virulence of HIV is the severity of the HIV, that infection. How severe is the condition? Somebody is asking, is it true most viral conditions are intentionally created? Viruses are not cells, and viruses are present everywhere. It depends on the condition. So some viruses can survive other conditions, some do not. But the ones that usually cause infections, they don't survive harsh conditions. That's why outside the host, they are easily inactivated. So they are not intentionally created. Another question from Stephen. With the opportunistic pathogens, is it that they can't cause infection? Yes, when the immune system is not weak, they don't take the chance to cause infection. They only wait for the right time, the opportunity when the immune system is down, or you have taken some medication and this is controlling the system, or some medication are preventing other cellular activities from going on. So they can take advantage of these conditions and then bypass the barriers where they are supposed to be limited and then overgrow and cause infections. So in a healthy person, these naturally will not cause infections unless they find themselves in other sites within the same host. But in the same site, they would not cause infections. Stephen, are you okay? Yes, Augustine.
Sir,
Yes.
sir, please. Good evening.
Good evening.
Sir, please. I have, with regards to the virus, I had a patient that was having a hepatitis B, and we tried to manage it, but we couldn't. But later on, he went to a herbalist, and the herbalist was able to give him some drugs, and it really worked because when he came back and test, it was negative. He went for the B profile, everything was fine. Now, in your note, you said the virus don't die, but they are activated. If you could explain further with regards to what the scenario I just gave. I don't know whether. Hello, sir. Hello.
Yes, they are inactivated. Yes, I can hear you.
Yes. Yes. So
yes, I can hear you.
Yes.
Okay.
Yes. With
Yes.
the scenario I gave.
So,
with the scenario I gave, now the virus don't die, but when the guy went, he was able to get his cure, and he's now fine. So does that mean that the virus is still there in his system?
Okay. So viruses are intracellular. They live within the cell to multiply. Without a living cell, they cannot multiply.
Okay.
So in your case,
yes,
because they don't have cells, they are not living organisms. That's why. So if you are not living, you cannot die. That is what I'm referring to. So if they are not living organisms, you don't say they have died. They have been inactivated. So in your case, the medicine that person took, it could be that there was an active compound that inhibits one of the pathways through which the virus uses to multiply. There are several pathways. So when you get to the viral life cycle and then the antiviral medications, we shall look at some of the pathways that they use to overcome the immune system or to multiply or replicate. So it could be that the medicine that the person took, it had an active compound that was able to block one of the pathways through which the virus uses to multiply or hijack the cell. So if that is blocked, the immune system will then have the power. So it's just like so once you take in the medication, that's an intervention. So that intervention will then stop the viruses, those invaders, from acting. So, and then release your soldiers, the ones that are able to fight the viruses. Once the viruses are then blocked and your soldiers are released, they become empowered to control them this time. So this is what probably the active compound within the herbal preparation was doing. So once the soldiers are activated, they multiply in numbers and then control the viruses so that they don't infect new cells. Once they are not able to infect new cells, their numbers will go down. They are broken down. Some are digested so that the proteins, the other molecules that are present in them can also be reused by the system. because they are also molecules, macromolecules. That's why you don't have the load coming up. So if you test and then you are not seeing the viruses, it means that the load has been drastically reduced and your immune system has taken control to monitor them. Now, immune system knows how they look like. So anytime the numbers are getting up, they know the weapons to administer to control them. So this is one of the reasons why the viral load will go down so that the immune system can take over. All right, so there's another hand up. We lost that person. So yes, Emmanuel.
Hello, sir.
Yes.
Yeah, so for the scenario that our friend also made, it also depends on the amount or the quantity of the virus at that particular time in the patient's blood or in the patient's system. So maybe it wasn't, or it also
Exactly.
depended on the stage of the disease or the stage of the disease the patient was. If it has been chronic, whereby the virus has
Yes.
replicated and then has taken over the patient's system. That one, no drug or nothing can be done. You just reduce it to a particular stage or a particular volume, and then maybe the patient will stop having other signs and symptoms. But it doesn't necessarily mean that it is gone forever. The particular scenario that you raised, if the patient stops taking that medication, the virus can replicate and then get back.
Come back.
Yes. So I
Yes.
also wanted to let you know that it also depended on the stage that the patient was, or the state that the virus had gone at that particular moment. Thank you, sir.
Yes. Yes. Thank you. So we have what you call limit of detection. Sometimes you do the test and it will show negative. Doesn't mean it's not present. It's the limit that your detection system can go to show that it is there or not. So as you said, if the numbers rise, it can then come back to detectable range. And when we get to the viral life cycle, hello, please mute if you don't have any contribution. So some of the viruses can also go into dormancy for some time and then come back in full capacity. They're also strategic. So let us not always assume that they are done or they are finally inactivated. So just like you said, it depends on the stage, depends on the viral load at that particular point in time. So if they are not monitored for some time, the patient who thinks he has been fully cured may suffer. So periodically he has to be checking the presence of these viruses. All right, thank you. So we have, yes, Finoria was asking, they block what? So there are pathways to work or take control over the cell. They follow certain paths or they use some mechanisms. So if you are able to provide an intervention that will stop the virus from moving to the next stage, you successfully control them and they don't infect new cells. So there are several enzymes that the virus will have. Once you control some of these enzymes in the viruses, you are on top of the game. You have the viral load coming down. All right. So if they cannot replicate, they cannot multiply. This is what it means. Comfort. They replicate to increase their numbers because they come with their DNA or RNA, and because they want to express their DNA or their RNA, the genes within them, if they can fully express them, they need to make several copies. That's what we refer to as replication. So if they are not able to make several copies, your cell can take over. Your immune system can take over because the numbers are too low for the virus to take control. Depends on the type of virus anyway. So once you control replication, the numbers cannot go up, and the immune system can be boosted to deal with them. So when they are inactivated and that matter, they can't replicate. Can they be cleared from the system? Yes, sometimes, like I said, you have what you call lysozymes. Lysozymes can digest some of these viruses so that the molecules that are present, they are made up of proteins. So that way in the system, covered up and then excreted. All right. All right, so let's move on. So becoming established after going through the entry point, then it can be through the skin. So when they get incision through the gastrointestinal tract, when food is contaminated and is taken in, water is contaminated and is taken in. So usually, once you monitor these entry points and you prevent them from getting into your system, you are okay to go because you don't know the type of organism that is entering. So once you check the food, the drinks, the environment, what you are inhaling, when it comes to urogenital tract, sexual intercourse, displaced organisms, so especially the females, if you use the washroom, how you clean yourself up, you can have some of the microorganisms from the anus into the vagina, so they can be displaced organisms. So once you check all these environments, you can control them from becoming established in the first place without even getting to this stage, the adhesion stage. Once they get to that adhesion stage, your immune system has to take over. So usually you can prevent them from the initial stage, and then you are all right. Some of them could be transplacenta, so from the mother to the baby. Please mute if you don't have a question. So we talked about the endogenous agents, those normal flora that are already present and then ended up causing infections. Exogenous is when you have those from the external part of the host. So they can come through the food contaminated, or drink contaminated, respiratory tracts. You have people who have the infection, drop infection could also occur. So if the organism was not originally present within the body, but it's coming from outside, that is exogenous infection. So that organism is an exogenous agent. But not all of them will end up causing infection because the immune system can control them at some point. So usually you can have exposure, but it depends on the number of organisms or viruses that you are exposed to. The numbers differ. So the minimum that will end up in an infection in a normal adult, so a healthy adult who has been exposed. So let's go back to this. You get exposed there's adhesion, there's invasion. Your immune system tries to control. So let's say you're exposed to 100, then 2 of them successfully place mute. Wait, that. So you're exposed to 100, then your immune system was able to control 98. 2 of them were successfully able to get to other sites and then multiply. And then those 2, they multiplied, and then now you have about 64. If for that organism, the 64 is the minimum that can achieve infection, that becomes the infectious dose for that particular organism. So the minimum number of those organisms or the agent, in the case of the virus, that can establish in a normal adult, which is the host, to result in an infection. That's the infectious dose or the ID. So if the infectious dose for that organism is small, that means they have greater virulence. The degree of severity will be higher, because even in small numbers, they are causing harm. So if they multiply, you can see how severe that disease would be. If there is no infectious dose, there is no infection, because you've not getting to the threshold. So if the infectious dose is 200 and you are having 100 or 150, infection will not occur unless they multiply and get to that threshold. But you don't have a counter in your system to be monitoring how many you have at a particular point in time before you try to take medication. So usually prevention is the best option. Don't wait till it gets worse. And the infectious dose may also change, especially if the immune system is compromised. So that's why we are referring to a normal adult host. So you can have these diseases or infections. The infectious doses, that's why I don't joke with that. So usually for the epidemiologist or the public health expert, they also pay attention to the infectious doses of these viruses or as degree of pathogenicity. So if there's an outbreak, assuming we have an outbreak, and then they are looking at the severity of that disease together with the infectious dose. So if the infectious dose is smaller, that means it can contribute to the severity. So attention will be given to that. So Q fever or query fever, 1 to 10 is enough to establish infection. For gonorrhea, about 1,000, typhoid, 10,000, need 10 million bacterial cells to establish an infection. But some of these organisms are also strategic or smart. They multiply at a very fast rate to get to that threshold. So we have a question from Maxwell. Why small infectious dose causes greater virulence? Okay, so let's go back. If the organism has one, just one can achieve infection, and you have 10 in your system, usually infection will progress to disease. So if you have one and is able to achieve infection, when you have 10 10 can make the condition more severe. Compared to this particular one, cholera, you need about 100 million cells. So 100 million cells, less than that may not achieve infection. So if you are exposed to one Vibrio cholera, somebody is exposed to one measles virus, smaller infectious dose will lead to infection. Infection will progress to disease. So the severity with a smaller infectious dose will be higher compared to one that needs of that particular kind of organism. You sorry, I lost connection. Please, can you hear me now?
Yes, sir.
All right. So, Maxwell, are you okay? Is your question answered? All right, let's move on. So, once you get exposed, they need to attach or adhere to the host and then penetrate. Tell molecules on the host. You know, you have some small pores within the host cells. So these can easily enter, but they need to firmly attach by way of these structures. So some have fimbriae, flagellum or flagella. Some can use a flagella for movement, but some of them will use this for attachment as well. Some of them will have slime layers or capsules that they can use to firmly attach to the intestinal walls so that they are not washed down, or they can avoid phagocytosis by the defense system. Some of them, like the viruses, will have spikes so that they can firmly bind to the host cells or the receptor cells of the host and then gain entry. Some of them will have cilia, suckers, hooks, or barbs for attachment to the host. So, for example, the bacterial cells, they have appendages like the that they can use to attach, and then they finally gain entry. Will release some toxins or open up so that they can enter the host cells. Some of them will have capsules. These capsules cover the cell walls of the bacteria, and they are slimy. So assuming the bacterial cell, the system uses some proteins on the surface of the cell to attack them. Once they cover themselves up with these capsules, the immune system cannot recognize them. So that's another way of avoiding being seen by the immune system and then being attacked. Once the capsules, and then they begin to invade other. Some of the viruses will use the spike of the proteins to attach and then enter. Once they enter, then they uncoat, they remove their capsids, and then they have the DNA or RNA to express what the virus has introduced. So this is how they enter, and then they remove the membrane and the capsid. This mutes. So once they are within the host, they have to find ways to survive. And the immune system will try to attack, sometimes by phagocytosis, engulfing those foreign materials or foreign agents, digest them with the help of some lysozymes, and or hand over them to other cells to kill them. So we can have blood cells. But these organisms are also strategic. They can also produce anti-phagocytic factors like the capsule, or they can release some chemicals, toxins, or vaccines that will neutralize what the immune system is trying to use to attack. Some of them will produce toxins that can neutralize the white blood cells because these are related to defense. So streptococcus, they can produce the leukocidins, which are toxic to white blood cells. And once they overcome them, they can bypass the defense system and then move to other sites whilst they are multiplying. Slime layer or capsule. So these are referred to as the glycocalyx. They can be slime layer or capsules with they can use to firmly attach to the host cell, cover themselves up, and then avoid being swallowed up or engulfed because of the slimy nature. Once they survive, they move to other sites and they multiply. Then once they multiply, we say there is an infection. Then they establish themselves and cause damage to tissues. And this becomes severe. So they will move from the infection to disease. So the virulence has to do with the severity of the disease after they have fully established themselves. So they have moved from infection, and now they are causing damages to the tissues, the organs. This is where you are feeling the symptoms. So how severe those damages occur, that's what we refer to as the virulence, and it has to do with the factors that they possess. Can be the characteristics or the structures. Some of them will cause diseases by dissolving some barriers in the host. So they can produce enzymes external to the cells themselves. So the pathogen will produce some enzymes outside its body or outside its cell to the host to dissolve certain barriers so that they can easily penetrate or move freely within those particular cells and tissues. Some will produce exotoxins. Like I said, these endotoxins are usually not released. So in the course of invasion, the immune system, some of them are able to kill in the case of the microorganism, or inactivate in the case of the viruses. And usually in the microorganisms like the bacteria, they get opened up, they get lysed, and finally they release these toxins. Some of these toxins, although the bacteria is dead, these toxins are still within the host, and they can elicit some responses within the host. Some of them can lyse, damage, or cause impairment within the host if they are in large numbers. Or sometimes, if you want to detect the presence of certain organisms, we can use the toxins that they have produced to detect them. But your body has a way of detoxifying when it gets into these organisms. So with time, it clears these toxins so that your cells cannot die or will not die. So examples of the toxins that can be released, the exotoxins, they are produced within the organism, but they are released outside the organism within the host cell, and they target specific sites within the host cell. Some of them can cause hemolysis. So, for example, the staphylococcus, they can produce these exotoxins, cause lysis in the blood, and we can have those that are produced by the sugar toxin, the cholera, the diphtheria. These toxins are also active, and they are also helpful in binding to molecules. So usually we call them AB toxins. So once they are able to activate enzymes, they are able to also bind to molecules and then move within the host defense freely. Yes, Augustine.
Sir, please, I wanted to know, with regards to the toxins, is it when they are
Okay.
multiplying that they release the toxins, or when they are excreting that they release the toxins?
Okay, so we have metabolic waste. So that is one. When they are, all organisms, when they feed, they will produce waste materials. That is one. But these toxins are released specifically to also fight. So they also have to fight back. So it is not as a result of after feeding or metabolic activities, the way that they produce, the waste products. But these ones are toxins that they are specifically producing to defend themselves. These exotoxins. So if it has to do with blood infection, if they're able to cause hemolysis, in a way, they can get nutrients, proteins from hemolysis. In a way, they can also cause the levels of the blood to go down. So these are specific molecules that they release to achieve coating. I hope it's clear. Augustine.
Sir,
All right, let's continue.
yes.
So the endotoxins, hello.
Sir, you go back to that slide. Is that slide that is confusing me? Maybe that's what I'm not understanding. That toxigenesis.
This one.
Ah,
Or which
thank
one?
you. That one.
Okay.
You see the toxigenesis, it says capacity to
Okay.
produce toxin at the site of multiplication. So I was asking that, is it when they are reproducing their kinds that they release the toxins, or when they are excreting that they release the toxins? That was what I was asking.
Oh, okay. So this refers to when you say at the site of multiplication, and take note, site of multiplication. Let's go back. here. When they are multiplying, it's because they want to get to a target within the host. So sometimes to get to the target, they need to produce some toxins. Some of these toxins will help them to dissolve certain barriers. So to dissolve certain barriers so that they can easily penetrate to those sites when they are multiplying. Because if the numbers are low, they may not achieve the capacity to dissolve those sites. So it's when they multiply and then become many that will enable the toxins, the chemicals they have, the threshold to dissolve those sites, and then they can move to the target sites and then achieve infection. So this is not about when they are multiplying and they are eating or they are feeding to release metabolic waste. This one, they are producing the toxins to neutralize the defense system or like they penetrate, or they need more nutrients. So they produce these toxins to lyse cells to break certain cells or more. Augustine.
Yes, sir. Yes, sir.
Okay. So the sites, endotoxins are not secreted. So usually it's when the organism, usually the bacteria, is damaged or killed. That's when, so it has to do with cellular damage within the organism, the bacteria. But these toxins are also not beneficial to the host cell. So they have to be neutralized. So the host will have ways of detoxifying these molecules that are produced by the cells. You know, when the organisms are multiplying, some will die. So in the course of dying, these endotoxins are also released. Please mute if you don't have a question. Amanda. So usually the endotoxins that are not released are lipopolysaccharides, and usually found in gram-negative bacteria, the cell walls of the gram-negative bacteria. I remember I thought about gram-negative, gram-positive. Or was it not this class? Gram-negative bacteria, gram-positive bacteria. How do you understand gram-positive, gram-negative bacteria? How do you understand these terms? Haven't I explained before? Okay, so when we talk of gram-negative or gram-positive, it has nothing to do with charge. Gram is the name of the scientist who developed the technique for staining the bacteria cells. So bacteria cells, all right, so it says yes, they retain a purple color. Yes, so I've explained before. Thank you. But we shall come back to bacteria staining and then we shall go deeper into lamb staining, so don't worry. Thank you. So for infection and disease, for the process can be broken into 4 distinct stages. We can have incubation, and the time for incubation varies, depends on the kind of organism that you're exposed to or the agent. So you can be exposed to an organism or infectious agent in the case of the virus, and then there could be some symptoms that are non-specific. No damage to the organs or tissues. But this can happen or occur several hours, several weeks, some months, or several years, depending on the organism. Yes, Finoria.
Sir, please,
Hello.
thank you. Sir, I'm sorry to ask this, but I kept wondering about what it may be. Sir, with regards to the infection dose, usually when you do without, they write some numbers for you. And those numbers, does it have something to do with the infectious dose, say?
Okay, so that is an estimation of the load, the load that they have counted. That is not the infectious dose. So assuming they counted 50,000 per microliter of blood, 50,000 per microliter of blood, you don't have just 1 microliter of blood in your system. You have several milliliters. So for every microliter, the number that they have written, that's the number. So assuming is 1,000. So if you take 1 microliter of blood, you have 1,000 of those bacteria or viruses. And you know 1,000 microliters is 1 milliliter. So if you multiply by 1,000, you are getting the number per milliliter. So you can imagine the volume of blood you have in your system and then how many you are getting. But the infectious dose is the minimum number of the organisms that they have estimated per microliter and the load. The minimum that can achieve infection. So if let's go back to this one. Let's say, oh, sorry, typhoid fever. The minimum they need to enter your system to cause infection, assuming you were exposed to 100, you didn't have the infection, but they kept multiplying. The immune system was fighting. They kept multiplying, binary fission. So 100 will double to 200, double 400, double 800, and so on and so forth. Once it gets to the threshold, 1,000, 10,000, sorry, the infection is said to have occurred. This is when you begin to feel the symptoms proper, or the immune system can no longer take control because they have exceeded the limit. And it varies for these organisms or viruses. But if they are counting the number you have in the patient, already it has exceeded the threshold. So it keeps multiplying. And they want to know the load, the bacterial load or the viral load per microliter. So it has nothing to do with the infectious dose. There has been an infection already, but they are checking how many you have at that particular point in time. they can also know the medication to give. Because if these levels are high, they could be associated with organ damage or disruption of the tissue function, or the load of the medication that they have to start giving. So once they know the load, they will know how to administer these medications. I hope it's clear, Finoria. They also use it to monitor
Yes, sir, but
the,
I don't
the
know whether
treatment.
you. Sir, I don't know whether you said this and I didn't hear it. But then,
Hello?
hello, sir. Hello, sir.
Hello?
Sir, please, can you hear me?
I can hear you. Yes, I can hear you.
All right, sir. So I was saying I didn't know whether you
Yes, I can hear
mentioned
you.
it and I didn't hear you. But then, sir, so does it mean it all boils down to the immunity again, that you have some people having, for instance, the way that somebody will have like 1/20 and 1/60 HNS, and then the person is having infection. So let me use the diagram we have on our screen here. For instance, the infectious dose for typhoid, let me use the typhoid, is 10,000. But then somebody with a weaker immune system with only 5,000, the person can still be infectious, or it must be 10,000 before it can cause an infection.
Okay. So for infectious dose, we are looking at a normal adult, a healthy adult who has been exposed. So on the average, we are focusing on the healthy one who has been exposed. How many is the minimum that can achieve infection? So if you are exposed to 10,000 for the typhoid, then that 10,000 is enough to establish infection. But if it's less than 10,000, that means they need to keep multiplying to get to the threshold and overcome the immune system, and infection would have said to be occurring. But if the immune system is compromised, the person may not reach the 10,000 because already the defense system that is needed to fight and maintain this below 10,000 is already low. So some 1,000 or 8,000 may be enough to establish infection. So once the immune system is down, the infectious dose will also come down. I hope it's clear now.
Yes, sir. Thank you.
Yeah, that's why some of the organisms will take advantage and become opportunistic, because those are already present in low numbers. Your body is keeping them in low numbers so that they don't establish themselves to become infectious. But once the immune system goes down, even at those levels, those low levels, they can result in infections. All right. So we've exhausted our time. Where were we? So let me finish with this slide and then we can end here. So it moves to the incubation stage, with the next stage where prodromal stage. So you still feel some discomfort, but non-specific. And sometimes people may not visit the hospital or will not seek treatment because one of the kinds of the discomforts that they've been experiencing, it comes and goes. And some people at this point may even be able to transfer the infectious agent to others, depends on the condition or the portal of exit. And then it gets to where they finally multiply in higher numbers at the target site. So here, when they finally get to their target site, this is where you get to the peak of the infection. So the prodromal stage is when they are trying to establish themselves in the specific site. Sometimes because the pH may not be conducive, they are trying to get to a particular site which will favor them. So they move from the prodromal stage to the actual state where the symptoms become very evident. The signs are showing that particular infection. And this is a point where people, because at the same time, will not even go to the hospital and then will see the infection going down. So some of them will relapse, become dormant, then come again, but this time be very severe. Some people will seek treatment. When they are feeling better, they will end the treatment. They will not continue. Then the levels will increase again and tissue or organ damage can set in. So the severity is seen at the period of invasion. The virulence is seen at the period of invasion. Then you begin to seek treatment. Person is recovering. So at this point, the point of invasion, the person can still be shedding the infectious agent, transmitting them, same as a convalescent period when they are getting better, responding to treatment. But people try to visit their relatives in the hospitals. You try to limit contact, but as Certain we don't agree. We don't understand why we shouldn't be allowed to see our patient when they are getting better. It's for their own good, but education is still needed in that regard. So some people are getting better and they feel like, oh, they need to stop the medication. Meanwhile, the numbers may still be present, and if you stop, they will multiply again and then activate the period of invasion. So these are the 4 stages when it comes to infection and disease. Any question up to this point? We've exceeded our time by 5 minutes. So try and go through the slides. I will have the slides. And if you can ask before we start the next lecture. If you don't have any question, I think we can end here. Enjoy your weekend.
Thank you, sir. Bye.
Thank you,
Thank
sir.
you.
Bye-bye.
Hey, I didn't log me out. Me that's the class. Na was here in point. I feel the airport.
I've been here.